the specific immune response. Before we begin, let's determine what this actually is and why we have developed this function in our body. By definition, the specific immune response is a bodily defense mechanism triggered by the presence of foreign substances, known as antigens. These can be found on the surface of pathogenic cells, those which cause disease, so it is imperative that our body is able to recognize them and, of course, destroy them. It all starts in the bone marrow, a stem cell-rich substance inside your bones that is responsible for the production of most of your body's blood cells, including all your red blood cells. It is here that stem cells begin their differentiation into a macrophage, by first turning into a myeloid precursor cell, then into a monocyte once in the bloodstream. Upon entering body tissue, monocytes mature into macrophages, which are phagocytic white blood cells. They can easily enter and exit body tissue by squeezing through gaps in the capillary walls. Upon detecting foreign pathogens, either due to chemicals released by them or damaged white blood cells, the macrophage engulfs them. It then releases digestive enzymes stored in its lysosome, which break down the pathogens. This process is known as phagocytosis. The remaining antigens are then presented on an MHC protein, also known as a major histocompatibility complex. This turns the macrophage into an antigen-presenting cell, or APC. Here we have a T helper cell. This helpful cell has CD4 receptors, which are specific to binding with certain shapes of antigen. The APC macrophage attaches to a T helper cell with complementary CD4 receptors. The T helper cell produces cytokines, small proteins which stimulate the T helper cell to clone into two different cells, both used in the secondary immune response. One of these is a T memory cell. This is long-lived and will quickly recognize the antigen if it ever returns, inducing a fast immune response. The second is an active T helper cell, which we'll talk about in a second. Here is a B cell. The black stumpy-looking things are called receptors. These bind to complementary antigens, which are then presented on an MHC protein, making this an antigen-presenting cell. This is where the activated T helper cell comes in. Those with complementary receptors bind to the antigen-presenting B cell and produce cytokines, which then stimulate the B cell to clone into two different cell types: B memory cells, which are long-lived and quickly recognize the specific antigen on return, and B effector cells, which differentiate into plasma cells. These cells produce antibodies, proteins which target the specific antigen and bind to it. Having antibodies clinging to you isn't the best thing for a pathogen. Not only do they make it harder to move and divide, they act as a label for passing macrophages. Macrophages have antibody receptors, which bind to antibodies, making it a lot quicker and easier to identify and engulf foreign antigens. Here we have an infected cell. This cell has been overloaded with pathogens and cannot destroy them on its own. It calls for help by presenting antigens from the pathogens on a CD4 protein, becoming an antigen-presenting cell. This is a T killer cell. Those with complementary receptors bind to the infected antigen-presenting cell. It then divides and differentiates into two clones: memory T killer cells, which are long-lived and will quickly recognize the specific antigen on return, and active T killer cells. These cells bind to the infected antigen-presenting cell and release chemicals that create pores in the infected cell, resulting in lysis. The infected cell dies, and so do the pathogens. That is the specific immune response. Not only does it destroy harmful, infectious, disease-causing pathogens trying to kill you from the inside out, but it also creates memory cells and antibodies to form a secondary immune response, one which makes these pathogens much quicker and easier to destroy if they ever decide to return to your body.